Okay, we're going to talk about some important components of skeletal muscle and skeletal muscle contraction. So I've drawn a few things on the board here. We'll start on this left side. So this is a sarcomere. This is really the material that's needed for muscle contraction. Kind of looks like a lot, so I'll go through it all really quickly. Um, you can see over here I have what look like these eights. These are going to be our myosin heads. These are going to be the active participant in muscle contraction. They're the thing that's actually going to be moving. They're going to be pulling along these. These are actin, F-actin. It's made up of a bunch of G-actin subunits linked together. And that's what it's going to pull on to shorten. So think of like if you're pulling on a rope, you're going to be shortening that end of the rope to this end of the rope. We have Z discs, these middle lines here, cap Z, that's what the titin connects to. Titin connects to the myosin. When the myosin releases from the actin, the titin will act as a spring and it will recoil. The muscle will be able to relax and lengthen out. What else do we have on here? Nebulin I drew down here. This is what the F-actin is wrapped around. We have these green Mickey Mouse shaped structures. This is going to be troponin. It's made up of 3 subunits: troponin I, troponin C, troponin T. And then we have this tropomyosin right here in red on this purple length of sarcomere here. So what happens, actin, it has binding sites on it that the myosin heads will bind to this actin, and then they'll have ATP and calcium come in, and that's what will propagate a muscle contraction. But these binding sites, they're actually covered right now by tropomyosin. And this tropomyosin won't let the myosin heads come in and bind to the actin. Also, we have this troponin. That's an important part here. The troponin I, that's going to bind to the actin as well. And then the tropomyosin, or the troponin T, is going to bind to the tropomyosin. When you have an increase in calcium, there's 2 binding sites. There's 4 binding sites on tropomyosin C for calcium. 2 are high affinity and 2 are low affinity. The 2 low affinity ones, they're going to be contained pretty much all the time. And what they do is they hold the 3 subunits of troponin together. The 2 low affinity calcium binding sites on troponin C, when they are occupied, they will cause a conformational shift in troponin, and it will actually pull tropomyosin off the active binding sites of actin, and it will allow myosin to come in. It'll grab, it'll bind to the active site, and it will be able to continue its process of muscle contraction. So those are kind of the main players in skeletal muscle contraction. So I'm going to come over here. I have drawn out a neuromuscular junction here. So if you remember from our last unit about action potential propagation, we have it coming down. This is an alpha motor neuron, a lower alpha motor neuron, so it's coming out of the ventral horn of the spinal cord. It's coming out. We have action potential, voltage gated channels. We have that are propagating our action potential down the axon. When it gets down close to this axon terminal here, we continue to have a high concentration of sodium outside, and it's going to want to come inside. We also have voltage gated calcium channels. Calcium also has a driving force that's going to push it into the cell. So we have calcium coming into the cell. If you remember, calcium, what it does is it activates snare proteins. It's the proteins on the axon terminal here, as well as these vesicles, and those are going to pull the vesicles to that synapse here, and it will fuse with the synapse and release its contents, which in this case is going to be acetylcholine. It's going to release acetylcholine into this neuromuscular junction. Acetylcholine is going to bind down here. This is ligand-gated sodium channels. Once again, we know sodium is going to want to go into the cell. It's going to further depolarize, or wants to depolarize and continue the action potential. One thing that's important, we have this little guy right here, this Pac-Man creature. He is going to take apart the acetyl and the choline. The acetyl is going to go away. The choline, our body's actually going to recycle. If I remember right, we get choline from vitamin B4. That's an important, essential vitamin. And that's going to get retaken into the presynaptic terminal here, and it's going to be recycled and reused for later. So that's the acetylcholinesterase, and that's a choline channel. The ligand channel, so we're continuing our action potential. It's going to depolarize this membrane. It's going to go up, and then it's going to come down, and it's going to go down these, what they're called T-tubules. Alright, the T-tubules, action potential is going down. We have another sodium, voltage-gated sodium channel here, so sodium is getting in. And then we get down here, and we get to a voltage-gated voltage-gated calcium channel, and this is called the DHP channel. And what it does is that it's a voltage-gated channel. It's going to open its door. However, external calcium, so calcium in the ECF, is not going to be the primary driver of muscle contractions. If you remember from biology in the cell, there's what's called an endoplasmic reticulum. Well, in the muscle we have a sarcoplasmic reticulum, which is the same thing, and it has high concentrations of calcium in the sarcoplasmic reticulum. And that's where we're going to get all of our calcium for muscle contractions, is from this sarcoplasmic reticulum. So this DHP channel, it opens up, and it actually doesn't really do anything besides stimulate this. It's called a ryanodine channel. This ryanodine channel is a mechanical gated channel, so you can think of the DHP causing the mechanical gate to open in this ryanodine channel. And when that opens, the calcium, which is always going to be close to the gates because it has this calsequestrin protein in the sarcoplasm that really attracts calcium. So calcium is going to congregate on this side close to that gate. When the gate opens, calcium is going to rush out into the cell, into the sarcomere. And if you remember what calcium does, we have lots of calcium. These low affinity binding sites on the troponin C are going to be bound, causing a conformational change, which is going to move the tropomyosin off of the active binding site of actin, allowing myosin to attach to the actin. So we'll come over here because I have that drawn right here. This is going to be a cycle, so this is muscle contraction in a skeletal muscle. So here we have where we left off over there. The ryanodine channel opened up. Calcium is coming in from the SR, the sarcoplasmic reticulum. So we have calcium coming in. So ADP is right here. So this is on the myosin head as well as an inorganic phosphate. Calcium's coming in. It's going to allow the myosin head to bind to the actin. Doing so, we know that when two things bind, it releases energy. So we get heat over here, but we also get, this is important, we also get a cleavage of this phosphate from this myosin head as well. When that happens, the phosphate comes off, the ADP comes off, and the myosin goes from a cocked position to an uncocked position, and that's what's called the power stroke. And that is how we get movement. So we have the power stroke, but the myosin head is still bound to the actin. So what do we have to do? We have to add ATP. We add ATP, and that's going to pull, that is going to give us energy to pull the myosin head off of the actin. And then through a process called hydrolysis, the ATP is going to be broken down into ADP and phosphate, and we have our cycle, and it starts all over again. Calcium, we have to remember that calcium is constantly, when it wants to be out of the sarcoplasmic reticulum, but we have channels in here. These are Circa pumps, sarcoplasmic endoreticulum calcium pumps that are constantly putting calcium back into the sarcoplasmic reticulum, and of course these cause ATP. They're one of our main consumers of ATP in this cycle. So that's skeletal muscle briefly, and then we'll talk just a little bit about some other, some smooth muscle contractions in just a second. Let me draw them on the board. Okay, I've drawn two cycles. You'll remember the skeletal cycle we just did, and then this is for smooth muscle. There's two different types of smooth muscle contractions. There is two different types of smooth muscle as well, multi-unit and single unit. Multi-unit is going to be connected via gap junctions, so it's going to send an action potential and it's going to be a wave motion. Peristalsis, I'm sure you've heard of it. That's the movement of your intestines. They kind of have that wave-like motion to propulse waste through them. And then tonics, think of things, these are going to be sphincters, urinary, and also like anal sphincters. You want them to be contracted until you need them to be relaxed. So we'll start here. This is basic, kind of the same thing as skeletal muscle, except calcium plays a little different role. We don't have tropomyosin in smooth muscle, so calcium, we still need it for contractions, but what calcium does, calcium is going to act on two different proteins. The first protein is calmodulin. Calmodulin, what it does is it is able to activate this myosin light chain kinase. That myosin light chain kinase is going to be able to force that myosin head onto the actin, and then it will leave a phosphate group on there as well. So we start with ADP up here as well as a phosphate. We have calcium, which activates the calmodulin. Calmodulin is able to activate and turn the MLCK with an ATP. It's going to attach the myosin head to the actin, and then the phosphate groups as well. With that, like we remember before, we have energy. The energy is going to cleave this phosphate. It's also going to have the ADP leave, so we have ADP coming off, the phosphate group coming off. We have this residual phosphate group on here, down here. This is the power stroke again, if you remember from skeletal muscle. But this is now in the uncocked position. So we add ATP. The ATP breaks a bond from myosin to the actin, and then we have ATP and that phosphate group still there. If the phosphate group is still there, we can skip this step and go directly over to step 2, and this would just be hydrolysis then. However, if there are continual high concentrations of calcium, it will make it so this MLCP protein will come in and it will cleave off this phosphate. If it cleaves off the phosphate, then we have to start all over again. So we'll come back up here with the ADP and the phosphate. You'll have hydrolysis up here. And then you'll have to start with the calcium, calmodulin, MLCK, ATP to get to your second step. And that's the phasic smooth muscle contraction. This tonic smooth muscle contraction is a little bit different. This is going to be something that stays contracted for a long time. I'm just going to edit this arrow here and I'll tell you why in just a second. So we have the same thing. So we have, here's our myosin head, our actin, we have our ADP and our phosphate. Calcium comes in, it's going to do the same thing. It's going to act on calmodulin, change its shape, activate MLCK, add an ATP, that's going to get phosphorylated, ADP, the P, and the phosphate, ADP, 2 phosphates. And then what's going to happen coming off here is we're going to have MLCP come in. draw that, MLCP. MLCP is going to come in, it's going to cut this bond, and the energy from here, just like before, when we have this, when it binds to the myosin head, we're going to have the cleavage of this phosphate and ADP, so we're going to have everything leave. We're going to have the ADP and 2 phosphates leave because the MLCP is here. So we're going to have a naked uncocked myosin head. We're going to add ATP to it, but because it doesn't have this phosphate group on there, ATP is going to have a hard time binding to the myosin head to release the myosin head from the actin. So if you think about it, you'll just have a contraction and it will just stick. It won't be able to let go. And this is called the latch state. This is why you have muscles, smooth muscles that can just contract continuously. And it just kind of gets stuck here until for some reason or another your ATP is able to bind. It's able to pull off the myosin head from the actin, and then you can get hydrolysis again. And it'll start the cycle over. Those are the 2 smooth muscle contractions. And then I will draw really quick, we'll draw some reflexes on the board, and then we'll call it good. Okay, we're going to talk a little bit about reflexes now. Here's some things on the board here, 3 different reflexes. We'll talk about muscle spindles. These are in your muscles, more towards the ligaments, and they measure stretch. You have 2 types that we're talking about first. In the muscles, we have the chain fibers and the bag fibers. The chain fibers, they have their nucleus spaced evenly throughout the fiber. These don't go the whole length of the muscle, but they are in the muscle. They do have a motor unit that goes to them, but they don't really contribute to force of contraction. Mainly what they do with the motor unit is that when a muscle contracts, they contract with it so that they can take an accurate measure of the tension of the muscle. We have 2 types. The chain fibers, those are going to tell us time of contraction. Those are going to be mainly for muscle tone. And then you have bag fibers, they're going to be rate of contraction. So if things contract quickly, your bag fiber is what's going to innervate that. Chain fibers, they have a type 2 nerve running from it, type 2, and then the bag fiber has a type 1A nerve. Both are myelinated. The type 1A is a little bit faster and a little bit bigger. We can look up here. So I have a brain stem drawn here, a cross section of a brain stem. They have use your hamstring muscle and then your quad muscle. So this is like, say, I'm trying to think of how to use it with a quad and the hamstring. I'll explain it, and then probably the best way to do it is like if you were falling asleep in class and you have your head coming down like you're nodding off, and then your muscle spindles are gonna feel that stretch in your neck. So you'll have one of your chain fibers, your muscle spindles, you'll have that afferent nerve pathway. So you'll feel it'll send it to the brain stem. It's gonna branch at the brain stem. So you'll get 2 effects. One will be an EPSP, so an excitatory response, and it'll go to that same muscle that he felt the stretch at, and that's going to pull the muscle back. But at the same time, we need to have an inhibitory response to the contra or the antagonist muscle to relax it, to allow you to bring your head back up, or to allow you to flex your gastrix so you don't fall if you're falling forward. Those are 2, that's the muscle spindle, that's a stretch reflex. You're going to activate the same muscle that's being stretched. We have another sensory organ, it's called the Golgi tendon body. It's in your tendons. What it's going to do, so it's going to regulate muscle contraction. If you have something in the gym, if you're trying to curl something that's too heavy, it's going to make it so we don't hurt ourselves. We don't tear a muscle, we don't avulse, get an avulsion fracture. So it's going to be up here in the tendon. It senses it, say, oh, that's too much. It's going to come down here. It's going to send one to the brain, and that's just purely for information because it's going to make the decision already. It's going to say, I need to send an inhibitory postsynaptic response to the agonist muscle and a excitatory response to the antagonist muscle. So if you think about it, these are really, they're just opposite of each other. They have a different organ that is sensing the information. The last one that we're going to talk about really quick is just the withdrawal and cross-extensor reflex. So say you're walking and you step on a tack. Your body's going to feel that. It's not going to be any of these organs. It's going to be some sort of receptor. You're going to have a pain receptor. You're going to feel there's a tack down there. That's going to come in here. It's going to synapse. You're going to have some interneurons. Interneurons are going to send responses to both legs. So you'll need an excitatory response and an inhibitory response to both legs. So in this one, we're going to send an excitatory response up here. This is going to be our quad muscle. Quad muscle is going to contract because we want to lift our leg off the pin. In order to do that, we need to have an inhibitory response to that hamstring muscle. So that's going to relax. We're going to lift our quad up. At the same time, if we do that, we would fall, right? So we need to put this down. So we're going to have an excitatory for our hamstring on the contralateral side, and an inhibitory for the quad muscle, so that we can switch from stepping on one foot to the other and picking that other foot off the ground, so we don't further injure our foot with the nail down here. Just a couple of things. So in order for us to have, so we have the response on any of these, if we are changing from a excitatory response, which any innervation stimulus is going to be an excitatory response because we're propagating an action potential, we need to have an interneuron if we are going to switch from a excitatory response to an inhibitory response. So you'll notice all these inhibitory responses are going to have an interneuron. The excitatory responses, they don't need one. They're going to stay the same. They're just going to keep on sending an excitatory response. So that's reflexes. One thing that I didn't cover, that I'll cover really quickly, just about muscles, is energy sources, where they come from. Let me get it pulled up here really quick. Energy sources for muscle. So I'm going to erase this really quick. I want this to end with this. So you have, from one of your energy sources, you have just systolic ATP. So this is going to be systolic ATP. I'll write that up here. If I have a good marker, I'll write it up here. Systolic ATP. You have the creatine phosphate pathway. You have glycolysis. And you have just aerobic respiration. So systolic ATP, this is just ATP that's found in the intracellular. It's available. You have probably about 5 to 6 seconds of cytosolic ATP. So what that's going to be for is quick, short things. If you're doing like a curl or doing like a short sprint, if you have to run for 5 or 6 seconds, that's going to be your systolic ATP. Creatine phosphate. So what that does is there's a creatine phosphate that is easier for the body to phosphorylize ATP from than to create it on its own, and it lasts for about 10 seconds. But it does require creatine kinase. So that's going to be the rate limiting factor for the creatine phosphate pathway. Glycolysis is the breakdown of glucose. It gives you about a minute of energy, and then after that we go into aerobic respiration, and this is going to be fatty acids. This is going to be what your marathon runners are using, is this aerobic aerobic respiration. All this up here is gonna be termed anaerobic. So this doesn't need oxygen, this needs oxygen down here. I think that's it for this time. We'll do another one next week.